Welcome to Health Professional Radio. I'm your host, Neil Howard. Glad that you could join us for another segment. In this segment, we're going to be speaking with Craig Parker. Craig is president and CEO of Surrozen. Now, Surrozen's a biotechnology company. He's joining us to talk about how the company is discovering and developing drug candidates to selectively modulate the Wingless integrated pathway, or WNT pathway. He's going to talk about the tissue-specific antibodies designed to engage the body's existing biological repair mechanisms with potential application across multiple disease areas. Welcome to Health Professional Radio. Craig Parker, thank you for joining.
Thanks for having me, Neil. It's my pleasure.
Well, as CEO of Surrozen, give us a brief look into your professional background and talk about your company's goal.
Absolutely. So I trained initially as a physician scientist and have worked in the industry for the last 20 plus years in various roles, from business roles to finance roles to research roles to clinical development roles. So came to Surrozen with a diverse background, which turned out to be really the perfect preparation for the diversity of therapeutic areas that we're exploring.
What is the Wingless integrated pathway?
So this is really one of the fundamental signal transduction pathways in biology. It's known to play a really critical role in embryonic development. The name Wingless came from the identification of the gene playing a role in the development of wings in Drosophila and fruit flies. Integrated came from a separate discovery of the same gene related to the integration of a particular a viral gene into the mouse genome, and this gene was expressed when that was integrated. So it turned out those were the same discovery, and so they were combined into this portmanteau of Wnt. So Wnt plays this critical role in embryonic development, in renewing stem cells, in organizing tissue, and then was discovered later that it played this really critical role in how the body responds to injury and how many tissues are maintained. So this is a pathway that can stimulate proliferation of stem cells, renewal of stem cells, differentiation of those stem cells into different very specific cell types. And we're trying to take advantage of that physiologic role that this pathway plays in maintaining and restoring tissue to restore tissue in response to injury.
So what have been some of the hurdles that you've encountered in that endeavor?
You know, this pathway consists of many different proteins and receptors, and there's extensive crosstalk between those. And so it's been very difficult to engineer molecules that have drug-like properties that can also be tailored to be very tissue specific. So if you try to just make a recombinant Wnt protein, there are a number of biochemical reasons why that's difficult, but those don't have very good drug-like properties, and they can't be made to activate only a specific tissue. So discovery made by one of our founders, who's a Howard Hughes investigator at Stanford, really opened the door to discoveries that we made that enable us to copy the biological activity of Wnt, but to do so in a very targeted way. So we use these antibodies to target Wnt signaling to very specific tissues or cell types, but ultimately we're really recapitulating that normal physiologic biological activity of the Wnt pathway, but only in the target tissue, only in the diseased tissue.
Are there different types of molecules for specific tissues?
Yeah, there are really two aspects. That one is the tissue itself Wnt responsive? So not all cell types, not all tissues are Wnt responsive. There are many tissues that are terminally differentiated where you really can't proliferate the particular cell. But there's evidence based on discoveries over the last really 30 years identifying what tissues are Wnt responsive, and those are very diverse and numerous. You know, they range from intestine, liver, salivary gland, retina, lung, pancreas. So that's our starting point, is do we know the tissue's Wnt responsive? And then we have these technologies that are quite flexible. So it's typically a different antibody for different tissues, but the format of the antibodies is the same, or at least as of today is the same. We have some future innovations that really expand on those formats that we've developed. So we can take this one format and adapt it to target different tissues. And we do that by substituting different binders in the binding regions of the antibody.
Does it make a difference how the tissue is damaged?
It does. Cells that we're targeting need to be the damaged cells. Yes, not every tissue would be amenable to this approach. But if that damaged cell type is one of these Wnt responsive cell types, or if restoring some of these Wnt responsive cell types can lead to restoration of some of the other features of the disease. So I'll give you an example of that. We've done studies in injury models in rodents of liver injury. And in those, hepatocytes are killed in many of these liver injury models, and as they are in humans. For alcohol injury, for example, kills hepatocytes. So you can replicate that type of injury in a mouse, and we've shown that we can really rapidly proliferate mature hepatocytes and restore liver function. You can also look at liver injury models where there's scarring or fibrosis. And this is what happens in liver diseases like NASH, non-alcoholic steatohepatitis. The liver becomes inflamed, and then it becomes scarred and fibrotic, and that leads to cirrhosis. Now, we don't have a direct effect, we don't think, on that scarring or that fibrosis, but what we've seen in rodent models of liver injury is that we can reduce that fibrosis. And we think that's a result of restoring normal, mature, functional hepatocytes. And that process of growing or restoring normal, mature, functional hepatocytes leads to a reduction in fibrosis. So we can indirectly potentially impact tissues that are diseased, even if the actual cell we're targeting isn't that specific feature of the disease.
SZN 1326 and SZN 043. These are two compounds that are for specific ailments.
Sure. I mentioned just a little bit about the first of those. SZN 043 actually mimics the activity of a different regulator of Wnt signaling. It's called R-spondin. That's a known protein in the body that's involved in Wnt signaling. And we've created an antibody that mimics the biology of R-spondin, but it targets it specifically to hepatocytes. So in multiple injury models in rodents, we've shown that we can target just hepatocytes. We don't activate Wnt signaling in any other tissues or any other cell types in the liver. And that leads to this proliferation of mature hepatocytes. It's important that they're mature. If they're immature, they may not restore liver function, but we've shown that we restore liver function and, again, can potentially reduce fibrosis. So we're taking that molecule into the clinic this year in 2022, and ultimately in a phase 1b, we anticipate taking it into severe alcoholic hepatitis patients who, as I mentioned, have as a feature of the disease this rapid loss of hepatocytes, and we think we can restore those. SZN 1326 is targeted to the intestine, and this molecule is mimicking the activity of the protein we've been talking about, Wnt. It targets a specific frizzled receptor involved in Wnt signaling called frizzled-5. Frizzled is the nomenclature for the receptors for the Wnt proteins. And we've shown in animal models of colitis that in this case, we can rapidly renew stem cells in the intestinal epithelium and expand all of the necessary cell types in the intestinal epithelium so that it's very normal looking and functional, and in the animal models improves both the barrier function that the intestine serves and also can improve the disease activity in these animals. So as a result of administering the antibody to an animal that has a highly damaged colon, we're able to restore normal-looking colon intestinal epithelium and normal bowel function.
Well, give us a website where our listeners can learn more, if you would.
Absolutely. So it's www.surrozen, surrozen.com, and you'll find information on the Wnt pathway and the company and the many publications of ours actually over the last several years. So we've published several scientific publications and quite a few abstracts at medical conferences that include some of the data that I've referred to from these preclinical studies.
Well, I thank you for joining us here on Health Professional Radio, giving us a little bit of your time. Thank you so much for joining us.
Thanks for having me.
You've been listening to Health Professional Radio. I'm your host, Neil Howard, in conversation with Craig Parker, President and CEO of Surrozen. Audio copies of this program are available at hpr.fm and healthprofessionalradio.com.au. You can also subscribe to the podcast on iTunes, listen in, download at SoundCloud, and be sure and subscribe to our YouTube channel at youtube.com, Health Professional Radio.